to make you move to the white chairs for next week. So fill in the white chairs for me, please. I'm sorry, I know. It's not supposed to be true. White chairs.
Come on.
Alright. Fill it up in the white chair. I don't know. I guess that's up to your teacher. Whoever your teacher is. All right. Okay. We're going to go ahead and get started. So let me have your attention up front. There'll still be people probably trickling in, but I will go ahead and start by introducing myself. So I know many of you, but not all of you. Hopefully someday I'll see some others in classes of my own. But I am a science teacher and I mostly teach bio classes. My name is Karla. And the reason I put this piece up here is just so that you're aware of my background. So I'm a veterinarian. I'm also a scientist that has a PhD in infectious diseases with an emphasis studying viruses. So that will come up today in a couple places. And really I'm telling you that because I'm a resource for you. I'm here at George School. You're here for like maybe 4 years for some of you still. And we should have a chat sometime if any of those things interest you. All right? Today, however, we are talking about epidemiology. And I know in AP Human Geography, you've already done a unit that touches on epidemiology, and we're going to start with a little review. So, who remembers the epidemiologic transitions? Yes? Tell me one. If you can give it to me in order, that's great. But not. Okay. Good. Okay. Okay. So we have a few examples. Yes, in the back.
The first is like famine and pretty inconsistent. It's a pretty inconsistent natural increase. I mean, crude death rate and crude birth rate.
Okay. For demographic transitions. Yes.
Okay. So the first one is like famine and pestilence, and the second one is like a revolution in medical technology that leads to like a decrease in death. And the third one is like obesity, and the last one is like longer life diseases like diabetes and kids and things like that.
Okay, very good. So we're getting there. So there were originally 3 and then 4 stages of this epidemiologic transition, and now there might be a 5th, which was touched on at a later point in your book. But yeah, we got a lot of these, right? So originally, we had a lot of infectious diseases causing high death rates, in part, causing high death rates. As we developed some better sanitation and better medical practices, understanding of the germ theory of disease, we sort of had this receding trend in our pandemics. We don't have things like the Black Plague coming out as frequently and wiping out such a high percentage of the human population on Earth, or at least in Europe. And then, because we start to live longer, now we have these chronic diseases like obesity, like cardiovascular disease. So maybe people are dying more often from heart attacks. But we can prolong that now because we have medical technology interventions that allow us to extend life even when some of these degenerative diseases are present. And now there's sort of a debate. Are we in stage 5 of this transition now? We're starting to see what seems to be an increase in emerging and re-emerging infections, sometimes from things that we thought we had controlled, and we'll come back to that in a little bit. And so, maybe, not everybody's in agreement about this, but maybe we're in stage 5 in some areas today.
Could that be related to the Black Plague outbreak in Madagascar?
Right, so we're still seeing, so we have Yersinia pestis, which is the cause of the Black Death in Europe in the 1300s. We have that right here in the United States, out west. And we still sometimes see what's called spillover events, where animal reservoirs transmit that to humans and we still get cases. So yeah, there are still cases of these diseases that crop up now and then. But the question is, can we treat them and prevent death from them? And what is the mainstay of treatment of bacterial diseases? Yeah, antibiotics, right? You take those for bacterial infections, not viral infections, okay? But for a long time, we thought, oh, you know, we're not going to be killed off by bacterial infections anymore because of these antibiotics. But today, what do we have? We have superbugs, which are resistant to many, or sometimes, really scary, all of the antibiotics that we have today. And so the question is, can we keep pace and come up with new treatments, or are these pathogens maybe going to win and create another big, huge pandemic? And we'll talk about some of these terms in a moment. So first we have to define epidemiology. Generally, you can almost sort of define it as the study of public health. More specifically, it looks at the incidence, we're going to define that term soon, distribution and control of diseases. And it's not just diseases anymore. It started out really focused on infectious diseases and then other genetic and chronic diseases, but it's also about injuries, like how many people in the past year in the United States died from an accident between a bicycle and a car and they weren't wearing a helmet, versus those who survived and were wearing a helmet, right? So collecting that kind of data and analyzing that kind of data and determining whether or not wearing a bike helmet is actually a useful preventative measure, is also part of epidemiology these days. But my favorite is infectious diseases, so that's what we're going to talk about tonight. So I want to also emphasize epidemiology is like at the intersection of so many fields of study. And I apologize, I just threw up a bunch of examples that I brainstormed in no particular order or even grouping. They're just sort of how they came to me. So there are many others, I'm sure, that we could come up with. So if any of this tonight is interesting to you, there are many career paths that you could follow that could ultimately help you improve global health. Along those lines, when we talk about the interdisciplinary nature of epidemiology, I'd like to talk about this, because most people haven't heard about this yet, and I like to spread the word. So there's a movement, sort of, I think we could call it a movement, among health professionals and environmental scientists to try to come together, collaborate more, discuss more, problem solve together, and communicate under this umbrella of One Health. So this is One Health for individuals, humans, for animals, and for ecosystems. So up there somewhere, there's like vet medicine, there's human medicine, there's ecology, studying our environment, impacts of humans on that environment. So it's this triad, we all impact each other. And more today, we're seeing how important understanding those intersections is in actually solving the problems of today. And so One Health is a very important initiative you'll hopefully hear more about as you pursue your own education and careers. Who else is involved? So, who's heard of the WHO? FAO? Maybe a little less well known, but good, a few hands. So the UN has a bunch of agencies within its purview, and some of them, like the World Health Organization, like the Food and Agricultural Organization, are intended to allow for some international cooperation and coordination, especially for the WHO when it comes to an outbreak response. They're key in helping a bunch of countries come together to help one or several countries in a region experiencing the outbreak. Other intergovernmental organizations, does anybody know the OIE? I'd be very surprised. I am not even going to attempt to tell you what the French acronym stands for, it's the old name, but it now today is the World Organization for Animal Health. All right, so it's kind of an equivalent to the WHO, but with a focus on outbreaks in animal species, which may or may not have direct relevance for human health. In the United States, we have the CDC, which is actually the Centers for Disease Control and Prevention. They just don't put the P in the acronym. And we have a ton of other groups working at the federal, state, and local levels to keep us healthy and to respond quickly if there's an outbreak. And other countries have similar structures in place. But then there's things that are outside governmental control. So NGOs, what's MSF? Again, I have a terrible French pronunciation, so I'm not going to attempt it. But the English translation, Doctors Without Borders. Okay? So that's an example of a non-governmental organization that's very often present during an outbreak, bringing clinicians to the area to be first responders. One Health partnerships, we touched on that already. And then, of course, whatever medical personnel are already in the ground and the location of the outbreak, whatever facilities they have, that's what we're starting with. That's what we have to work with. And sometimes it's really strong, and other times it's fairly weak. And that is important to consider as we respond to the outbreak. Scientists, super important. They have to liaise with the clinicians, figure out the biology of the pathogen and how we might intervene to reduce cases and hopefully ultimately stop the outbreak. I put media here. That's not a typical thing on this list, but I feel very importantly, media plays a huge role in how the public responds to outbreaks. And so journalists, I think, have a very strong ethical obligation to correctly report and consider what their report is going to mean when they're reporting about something that has such sort of a high level of anxiety associated with it, or fear. And so journalists need to liaise with the scientists, liaise with the clinicians, understand, sort of be a specialist in reporting outcomes if that's what they're going to do, because they have the ability to either sort of stabilize an area or have it erupt into chaos, depending on how the reporting happens. All right, moving on. We need to do a little terminology. And all these slides will be available to you if you wanted to look back at them as teachers require you to know some of these terms. I'm probably throwing in a few new ones. These first three, if a disease is endemic, that means it's already present in an area. What is a disease that's already endemic here at George School? Do you think? Okay, so rhinovirus, probably there are many, hundreds of strains of rhinovirus, right, that cause the common cold. Yes, endemic to the area. Seasonal influenza virus. What about bird flu? Is that endemic in this community? No, let's hope not, right? That is going to raise red flags. We're going to have the CDC right here on our doorstep if we have bird flu here. But there's potential for it because of our connectedness with the world. So endemic means it's already here, it's already present. We see a baseline of cases throughout the year. Epidemic means it's new for a given location and a given population at a given time. And so it's spreading rapidly, it's spreading a little bit sort of broadly, but still we're talking, somewhat contained. It transitions to a pandemic once it reaches like multiple countries, certainly a whole continent, and then pandemic, a true pandemic, is worldwide. So the virus I studied when I was a PhD student was canine parvovirus. When it emerged in the 70s, it was a brand new virus, never before seen in dogs. It turns out it came from a virus of cats, but we didn't know that at the time. And it spread around the world in less than a year. That's pretty incredible. And now it's in the wildlife, so it emerged as a disease in domestic, like canines, so our pet dogs. But now it's spilled over into many different carnivore species around the world. Other diseases may have the potential to do that, and that's what scientists and health professionals are really concerned about with this potential stage 5 in our epidemiologic transition. And how can we prevent that pandemic from happening? And how can we respond to areas that have these outbreaks, these epidemics, to help them recover and then hopefully prevent a future outbreak? So zoonosis, reverse zoonosis. Zoonoses are some of the most common new emerging diseases. We'll talk about that in a minute today. And that means zoonosis means a disease is jumping, switching from some other animal species, a wildlife reservoir, domestic species, into humans. Reverse zoonosis is the opposite. So I know some people in the audience know this, but some of our great apes, lots of people love to travel and see our great apes in nature. But we put them at risk by doing so. There are many respiratory pathogens, for example, that we as ecotourists transfer to those animals. And they're endangered species, very low numbers in some cases, and they're getting sick from us, potentially reducing their numbers even further. So it goes both ways, and we're very, again, interconnected these days. And we need to keep that in mind and work together to try to prevent the spread of those, both in other animal species and in humans. Vectors and fomites. Vectors, generally, we refer to that term, although it's a little bit used loosely sometimes, but generally a vector is an organism, something that can transmit a pathogen between people. Can you think of an example? Okay, good. So rats could be a vector. We'll come back to that. Okay, fleas definitely can carry diseases between people. Another one. Mosquitoes, for sure. And ticks, that's a good one for this area. Mosquitoes, yeah, so ticks can carry Lyme disease in this area along with a number of other pathogens. Mosquitoes, maybe malaria is probably the most common And again, there's different species of mosquito. Different mosquitoes carry different pathogens. Some vectors can amplify their pathogen. That means the pathogen can replicate inside the vector. Others, it's just mechanical. It's just like, well, I, bit you. I have your blood in the body of the mosquito now, and it's being carried along with the pathogens that bring that blood over to the next person the mosquito bites. That's just mechanical transmission. Sometimes, though, it's being amplified as well. Fomites are non-living things. That would be like a door handle or a phone that has been sneezed or coughed on, and there's pathogens on that phone, and then that can transmit to the next person to pick up the object or touch the object. All right. And then finally, incidence versus prevalence. These are important terms with very precise definitions in epidemiology. Incidence is the number of new cases over some period of time, so you get a rate. The rate at which new cases are showing up in a population. Prevalence is like you just take a slice through the population and you count how many people are sick. Old and new cases both together. All right, so two different ways to get a sense of what is the impact of the disease on that given population. And then we have this idea of emerging and re-emerging diseases. So an emerging disease is something that we're seeing for the first time in a given population, in a given place, in a given species, humans, in our case. Re-emerging, it's things that have already been known to infect humans for a long time, like tuberculosis, which we'll come back to. But now maybe it is emerging in a new way. It's antibiotic resistant, for example. So it has new properties. So, in your book, there were a list of causes for these emerging and re-emerging pathogens. Why are they here today? Why do we maybe have a stage 5 present? And so, these big bullet points, those were in your book. And then I expanded on them a little bit. So, the evolution of pathogens and vectors, your book gave you examples like antibiotic resistance in bacteria. Like mosquitoes becoming resistant to the insecticide, and now we have more vectors around to transmit the pathogens that's present or endemic in an area, like malaria in tropical regions around the globe. I added in host switching. This is the phenomena that occurs because of this piece. We're more connected, but that means we're also having more contact. We're encroaching more on wildlife land. We're moving our domestic species right up next to the forest. And so humans, wildlife, domestic animals are sharing pathogens more. We get these events where we have spillover from some reservoir that's not in a non-human species into humans, and vice versa. So host switching has more chance of occurring because of that increased contact. And when host switching occurs, we have a virus like canine parvovirus that I used to study that suddenly can make use. It's changed just enough. And I studied the exact amino acid changes in its proteins that allowed it to now use a receptor in dogs instead of just cats. It wasn't many changes. And if we give enough chances for nature to just sort of pick and choose those mutations, what might work, we might get more and more emerging diseases. People are really afraid that this bird flu, so this highly pathogenic avian influenza virus that kills birds and people, if it gets into people, they're really worried that that virus eventually will figure out, just through random mutation and chance, a way to spread better between humans and become the next influenza pandemic that spreads worldwide, but it has a super high fatality rate. And that's sort of scary. But hopefully the scientists and clinicians will be ahead of the game and will be able to stop it in its tracks. That's the goal. Some of you might hope for that. All right. So we still have large areas around the globe with poverty, including here in the United States. When poverty is present, there could be less sanitary conditions, there can be less access to clean water, there can be less access to treatment if they get sick, and less access to vaccines and other preventative measures to keep them from getting sick. And so your book gives the example of tuberculosis. Tuberculosis has been around in people a long, long time. But we still have large regions where we don't even have the ability to diagnose the cases in certain areas and say, yes, you have TB. You should start this treatment so that you can get better and at least not shed anymore and not spread the bacteria to other patients. And so I'm going to talk again about TB in just a moment. But yes, the global movement of people have certainly contributed to this potential stage 5. So I have this little interjection. I think we have time because it's so very cool. This is one of my favorite NGOs. It's not very well known. Hero rats. Has anybody heard of hero rats? Okay, you'll have to, yes, you'll have to go check out the website. You can actually adopt these guys, which really means you're paying some money to this group in order to help them raise these rats to do the work that they do. So these are giant pouched rats. They're native to the African continent. This group, APOPO, that is an acronym for a Belgian company. And they have headquarters in Tanzania. They now have been at this about 20 years. Originally, they worked with these rats to train them to detect landmines. They're so light they don't set them off, so they can just go back and forth way faster than previous methods and scan land, identify where the landmines are so they can be properly inactivated, and we can return the land to use, like agricultural use in the community. Now they're also being used, their excellent sense of smell is also being used to scan like 100 samples. So a patient that might have TB comes to a clinic and coughs and gives a sample. These rats can find positive samples way faster than the traditional laboratory methods, making this, let them talk for a minute. Maybe not. So now the sound also goes over. Interesting. So I did see this before.
Standard method of detecting TB, microscopy at public clinics, is slow and precise. Every year, about 9 million people will contract tuberculosis. Of those, 3 million will not be diagnosed, and 1.5 million people will die. In fact, TB now kills more people than any other infectious disease. The standard method of detecting TB, microscopy at public clinics, is slow and imprecise, creating a vicious cycle of people with TB being sent home to infect others. To help with fight TB, Apopo deploys specially trained TB-detecting hero rats. They detect tuberculosis using their extraordinary sense of smell and are faster and more sensitive than microscopy. How does it work? TB suspect patients give sputum samples at clinic for examination, and one of these is sent to Apopo's Central City lab, where it will be tested within 24 hours. A hero rat quickly sniffs each sample. If it detects TB, it hovers over the sample for 3 seconds. Clinical report training using control samples helps ensure accuracy. Apopo technicians verify rat suspect samples, and if TB is indeed present, Apopo notifies the public clinic that the patient should start treatment immediately. Hero rats' specialized training makes them more efficient in microscopy. With global support, the hero rats would be a powerful tool for addressing TB at source by screening high-risk populations in factories, prisons, and high-density dwellings. Apopo is improving clinics' TB detection rates by over 40%. This is breaking the TB cycle, getting more people on treatment and saving lives.
Alright, so the point here is like, be creative, think outside the box, and you can help solve some of our world's problems as you move forward in your careers. So, let's see. Alright, so I'll put the categories up so you just stop me away. But, let's get some examples of pathogens that may or may not be emerging, may or may not be re-emerging, but I'm just going to toss some of these out there, right? Is everybody paying attention? Alright, so they're okay. So, read the tag, learn about your pathogen, and be ready to share in a moment. Heads out. Oh, right here. Gotta pay attention. Stop more. That's important for epidemiology considerations. Good. Dengue fever. Is that, what kind of pathogen is that? It's not a parasite. Not a bacteria. Virus. You'll soon find that most of what you have are viruses because that's my favorite. What do you have back there? What are we trying to do with polio? Anybody know? Eliminate it globally. Right? We're having challenges doing that because of conflict in areas where we still get polio virus outbreaks. But someday, what have we eradicated globally? Smallpox. Another one? Anybody know? Big pox. Rinderpest. It is an important pathogen of cattle, and we've eradicated that. All right, who has some bacteria? Yes. Lyme disease is a bacterium. Ooh, is that a bacteria? Giardia. Read the card. Is it a bacteria? It is a, open the card. It is a protozoan. So it's a parasite. It's a single-celled organism, eukaryotic organism. Yes. Salmonella. What is it? Good. Bacteria. What else? What do you have, Colin? Yeah. What is the pathogen? Can you read it? What kind of pathogen is it? Fasciitis. It causes necrotizing fasciitis, and the pathogen's name here is Streptococcus pyogenes. It's a bacterium. Yes. What do you have? Good. That's my only fungus that I have in my collection. Candida is a natural inhabitant. We all have Candida in our guts right now. The problem is Candida front. Shh. Up front. Attention here. I want to see eyes. Eyes. Well done. Thank you. Yes. All right. So we have multiple classes of pathogens. During an outbreak, we might not know, are we dealing with a bacterium, are we dealing with a virus, until we do some further testing. But once we know that, we have a sense of what we can use to try to combat or at least reduce the spread of that agent. So I took this snapshot from the health map yesterday, last night. Look at all of these notifications about outbreaks. Now some of these are foodborne illnesses, some of these are diseases just in animals, in marine life, but a lot of them are outbreaks in human populations. And these are reports about them. What do you think might be happening right here? Any guesses?
I can't think
You may or may not know. It is actually an ongoing Ebola outbreak. Yes. And we're going to talk about a very large Ebola outbreak towards the end of this talk. So, how do pathogens spread? There's a long list. I put some up in a moment, but let's brainstorm. How do pathogens spread? Yes?
Animals.
Okay.
Oh, that's what they were.
Okay, they can originate from animals, so they can spread from animals to humans. Be more specific. How do they spread? What controls how fast they spread?
Interaction stuff.
Okay, so how densely populated is the species in which the outbreak is happening? We're going to extend that. Yes? Okay, good. So how well is the species adapted to its environment, but also how well can it resist the infection? Are there some naturally immune individuals already in the population? What else? How can pathogens spread? What affects the rate of spread? What's that? Okay, yes, definitely. So now we're back to fomites. Fomites can spread pathogens. Yes? Okay, good. So that tells us where it might come from. These are the types of things I'm looking for. So what's the incubation period? How long after you're exposed until you show symptoms and shed the disease or shed the pathogen? How long are you infectious? Can you think of what's one of the longest infectious periods? What pathogen? Any ideas? HIV, yes. So, human immunodeficiency virus causes an infection in people, eventually leading to the syndrome called AIDS. And those individuals are infectious for the rest of their lives. So very long period. Versus some of these pathogens that we were just talking about, it's a short, acute, we call it, acute infection. It comes and goes. So the route of transmission, whether or not coughing and having airborne droplets spreads the virus, or do you have to be in really close contact with the infected individual? So there are some diseases in pigs that are known to spread like 2 miles on the wind. That's crazy, all right? If that happened in human populations, we would have a big, big problem. So hopefully someday we don't get that. But that's a concern. So again, density. How close are we all to each other? How susceptible are we? Are people immune? Are people vaccinated and have immunity against that infection? And do we have access to medical care? Can we get treated? Can we be removed from the population so that we're not infecting others while we're sick? And then all these different environmental factors play a role as well in terms of how well pathogens spread. So we can model how these diseases spread. So is anybody in Travis's computer science class? You're going to do this. You're going to learn how to code that model and be able to have different inputs in order to run it. I stole one from the internet, but you guys are going to do just that. So these different models can be very simple. You have a compartment. Some of you right here are susceptible maybe to influenza virus this year. Some of you have been vaccinated and you're actually already in this recovered pool because you're immune. And then others, hopefully not right now, are infectious or could become infectious. So we can calculate the rate of movement between those different compartments and build more and more complicated models to sort of predict how a certain outbreak is going to progress, and then figure out what interventions are going to work best at stopping it. That's really the goal. All right? And so again, mathematics, right? Computer science can come into this and play a role in helping us figure out how to control outbreaks. There's this idea of All right, and so I want us to get sort of a concept of this. It's called R0, and it can be mathematically sort of estimated from outbreak data. And each sort of pathogen has a unique R0. It's usually a range, but let's get a sense of this. So, Catherine, I'm going to have you stand up for me. We're going to give this pathogen that's infecting Catherine an R0 of 4. What that means is, well, she's infectious, she's going to infect 4 other people. So while you're standing there, without moving your feet, I want you to reach around, and I want you to tag gently 4 people. No moving, no moving, no moving. So tag 4 people. No. All right, 4 people who just got tagged, you stand up. Now you're infected, and you infect 4 more people. So tag 4 people. If you need to move a little to get your 4, you may. Tag people, stand up. People just tag. All right, take your seat again. So what do you think about that outbreak? Was it growing or subsiding? It was growing fairly quickly, all right? Let's try again. Chase, stand up for me. All right, we now, actually I want 2 people. Tim, you stand up too. So now we're going to have an R0 of half. That means only one of you, I'm going to pick Chase, one of you is actually going to infect one other person while you're sick. So tag 1 person. All right, you stand. Can you stand for me? What's your name? Your tag. You stand. And now it's a half. I'm going to say you infect 1 person. All right, stand for me. It's still a half. You don't infect anybody. What's happened? Yeah, it's died off. So what's the magic number? R0. We want it to be less than what? 1. We want R0 less than 1 to stop our outbreak. Let me take your seats. Thank you very much. So R0 is the number of individuals infected by a sick person over the course of their whole infective period. And we want R1 to get to less than 1. So we want to start interventions that will bring R0 less than 1 and stop the spread of the outbreak. So here are just a couple examples of R0 values. Ebola sounds scary when it's in the news, but look, it's one of the lower estimated R0s. Directly comparing between these is a little tricky. It's better to just think about, we need this number down below 1 to stop the outbreak. We need this number down below 1 to stop the outbreak. But still, it gives you a sense. Measles, I said I'd come back to it. One of the most contagious pathogens we know. Get vaccinated. Vaccination only helps our whole population if 90% of you are vaccinated because it's so contagious. That is why we're getting a reemergence of measles today. Because the population's vaccine protection has dropped below the coverage needed to prevent outbreaks of measles. Yes?
What's the difference between airborne and
So it's airborne droplets mean you cough. Have you ever seen those coughs on YouTube where it shows? So those droplets carry the particle, but they do eventually settle out on things that become fomites. But airborne is actually can be carried even further. So that would be like these diseases in pigs that can travel 2 miles. But even the measles, it causes a bunch of a rash and then some scabs, and you're shedding these particles into the air, and they can travel even further than droplet transmission. Lovely thought. So I'm going to skip this. This was actually just talking about R0 in the Contagion movie, but I will put in a little plug for it. It's a fun movie. The first half of it is they touch on a lot of real stuff, but in the context of something that's been a little distorted in terms of could this really happen. And then the second half is like a doomsday thing that just gets a little kind of crazy. But it's a good movie if you like that kind of movie. So here are the steps of an outbreak response. We don't need to worry about all these details. That's what epidemiologists do. That's what people at the CDC, the WHO, that's what they're taking care of during an outbreak. But there's a few key things I want you to understand about what they're doing. They have to define what a case is. You would think this is so easy. This is critical to be able to study any kind of outbreak. You need to know who are we calling sick with this disease causing the outbreak, and who is actually maybe sick with malaria instead of Ebola. So we need to discriminate between diseases that initially can have very similar presentations. So they have very precise case definitions that must be developed. It's easy if we identify the pathogen. If it's a new pathogen we've never seen before, this can be very tricky. Then we need to do contact tracing. And so I'm going to try again here. If it doesn't work, we'll skip it. But I do want it to work so bad. Let's just try. Alright, we'll just play it from here. Well, maybe. We're almost done.
The current Ebola outbreak is the largest Ebola outbreak in history, and the first ever in West Africa. At CDC, our mission is to prevent, detect, and stop disease outbreaks wherever they occur. That's why CDC disease detectives are hard at work 24/7 in West Africa.
Hi, Greg here. I'm with Ishmael, and we're going out with our UN colleagues to train contact tracers.
In the fight against Ebola, one of the most important tools we have to prevent spread is...
Okay, that girl is a classmate from Mexico. I was actually just telling her this story. So this is Alana Schaefer. I just visited her at the CDC this past summer. She's now working on leptospirosis and domestic cases. But she was part of the CDC response that got sent over to West Africa. And so she helped with contact tracing. What do you think?
In fact, it is the key to stopping the outbreak and saving lives.
She wanted to know how we are going on. Anybody who has been sick, anybody who has been in the hospital.
Contact tracing means finding everyone who comes in direct contact with a sick Ebola patient. We ask the Ebola patient or their family who the patient had contact with since they started their symptoms. These contacts are then found and watched for symptoms of sickness for 21 days to see if they become ill. If a contact begins to show symptoms of Ebola, he is immediately isolated, tested, and provided care, and the cycle starts again. All of the new patient's contacts must be found and watched for symptoms of sickness for 21 days to see if they become ill. The process is repeated until there are no new patients.
That's how we're going to stop the transmission chain of Ebola.
Contact tracing identifies new Ebola cases quickly, so they can be isolated as soon as they show symptoms, preventing spread to others. In the event someone with Ebola travels to another country, we would use contact tracing there as well. The truth is, even one missed contact can keep the outbreak going. But by carefully tracing all contacts and isolating new cases as soon as they develop symptoms, we can prevent further spread. By working together, CDC and its partners will stop the outbreak and save lives from this devastating disease.
I'm going to stop it there. So, very important. Contact tracing helps identify, and then we can quarantine and treat infected individuals and potentially infected individuals and stop the outbreak faster. But it takes a lot of resources, a lot of manpower. And who are the people who tend to get sick during these outbreaks in the chain of transmission? Family for sure, and one more big group. The doctors, the healthcare providers. So we need to keep that in mind. They go in with lots of training, but things happen and they do sometimes get exposed. So they're putting their life on the line. At least with Ebola, what's the fatality rate on average of Ebola outbreaks? It can be as high as 80 to 90%. On average, it's about 60%. So 2/3 of the people who get Ebola die. So that's a very high fatality rate, and it was very devastating in Western Africa. And if we had more time, we would go into why. One last thing, epidemic curves are super important tools for epidemiologists. You can go onto the CDC's website and track our influenza epidemic curve every year. So this is for last winter season, so from 2017 to 2018. And the different colors just represent different strains of the virus that they've tested for. But this tells us a lot about the course of the outbreak. Is it still rising, growing? Is it starting to subside? And then we need to think about what happens, and this, we'll just end on this note. We have to think about what happens in these areas after the outbreak has subsided. So Western Africa already had a very poor medical infrastructure. And then they got bombarded with Ebola. People who had TB, malaria, which are endemic in the area, they weren't getting the treatment, they were scared to go to the hospitals because Ebola patients were there. And so there is still long-term, ongoing rebuilding needed in those countries, even after the fact, and Ebola itself in the area has died out. And so that's important to keep in mind. There's a long recovery period in a lot of these cases for those countries. All right, thank you very much for your attention. Leave those microbes for me, please. Thank you very much. You too. You're Someone left their phone. I'm sorry.
It's so cute.
I know, right? Giant microbes. Yes. Yeah.
Oh my god, is there a video?
Stop
But